This is the third video on control of gene expression in eukaryotes. The first part was on epigenetic marks that can be put on the DNA that regulate whether a particular gene is transcribed or not. And this would involve marks that are put on the histones and also marks that are put on the DNA itself. Then the second part was the example of the GAL system. And this third part is going to be looking at post-transcriptional gene regulation in eukaryotes. And specifically, we're going to be focusing on RNA interference. So there are a number of ways that genes can be regulated, and we've kind of gone through some of these already. So we've talked about the regulation of transcription itself, and this involves these two levels of the regulation of the compaction of DNA and also whether you transcribe it or not. So I just want to mention a few of these mechanisms of post-transcriptional gene regulation. Splicing of RNA, of pre-messenger RNA, is one important way that genes can be regulated. Processing of messenger RNA is also something that is regulated. So this would be adding the poly A tail that can regulate RNA stability. And then there's various mechanisms of regulating whether that messenger RNA is translated. One of the big mechanisms in eukaryotes that regulate whether a messenger RNA is translated is something called RNA interference. And then finally, there are regulations of the protein itself once it is translated, things like phosphorylation. So RNA interference is specifically a regulation of messenger RNAs, and this is something really completely unexpected at the time that it was discovered. It is the ability of double-stranded RNA to lead to the degradation of messenger RNAs in the cell that have the very same sequence as that double-stranded RNA. As many as 30% of all human genes are affected by RNA interference. It's also been really important as a mechanism to experimentally regulate gene expression. So I want to tell you a little bit about the discovery of RNA interference. So as we talked about in prokaryotes, there's been this idea of antisense RNA that has been around for quite a while. And the idea here is that a RNA that is complementary to the normal messenger RNA, which is called antisense RNA, can bind to messenger RNAs and either block their translation or lead to degradation of that messenger RNA. And so people have been very interested in doing this experimentally so they can inhibit genes in cells. So inhibiting genes in living organisms has been a goal of experimental genetics for a long time. This is the primary way we learn what the function of a particular gene is. And people have tried this mechanism of antisense RNA, and they've only found limited success in eukaryotes. And so here's an example from way back in 1984, which is looking at a particular protein that is being expressed, shown by this dark stain. And people have tried these experiments to inhibit that particular protein by putting antisense RNA into those cells. And they really found some success with it, but not super success. So there's still a little bit of staining of that dark color in those cells. So along came the 90s, and two scientists named Andy Fire and Craig Mello decided to really try to work this out. And so what they did is they very carefully added different types of RNAs to an embryo, a nematode worm embryo called C. elegans. Now this is just a 4-cell embryo. It's pretty small at this point, but there's a whole bunch of messenger RNA that is in this cell, and it's shown by the dark stain. So they decided to test antisense RNA, and they saw a modest reduction in the total messenger RNA level, and that's seen by that modest amount of dark stain in those embryos. As a control in this experiment, they did this thing where they actually took the normal messenger RNA sequence, and antisense RNA, and they actually hybridized it together prior to injecting it into that embryo. And they were really surprised to see that that double-stranded RNA strongly inhibited all of the endogenous normal messenger RNA for that particular gene. And it was really an undetectable level of messenger RNA of that gene. Normally, it's found at very high levels. So they were completely surprised by this, and they looked into this a little bit more, and eventually they figured out that it was actually this completely novel mechanism that cells use to inhibit genes that have a complementary sequence to double-stranded RNAs that are found in the cell. This is Andy Fire and Craig Mello, and they actually won the Nobel Prize for this discovery in 2006. This was the very important paper that they published in 1998, which first described this strange effect that they were observing. So I'd like to tell you a little bit more about this. So certainly Fire and Mello did additional work on this, but then a lot of other individuals also contributed to discovering the pathway that is involved in the effect that was observed with double-stranded RNA. So RNA interference, it is the effect of double-stranded RNA to cause the degradation of similar messenger RNA sequences in the cell. So what was found is that double-stranded RNA is actually found to be bound by a particular protein called dicer. Dicer does just what you'd think it would do. It chops up that double-stranded RNA into these little pieces that are about 20-25 bases long. These are called short interfering RNAs. And then these short interfering RNAs are bound by a complex of proteins in the cytoplasm called the RISC complex. Now, RISC, what it does, it actually separates the two strands of that siRNA and uses one of them as a guide to look for similar sequences of other messenger RNAs in the cell. Once a similar sequence is found, it base pairs with that siRNA, and this base pairing, specific base pairing, leads to the degradation of that messenger RNA. Now, this was a pretty exciting finding for being able to experimentally inhibit genes in cells. And so people started using this right away. So they'd inject or give cells double-stranded RNA that had the sequence of the gene they wanted to inhibit, and then this double-stranded RNA was figured out to go into this RNA interference pathway. So dicer cuts it up into small siRNAs, and then the RISC complex binds it and uses one of the strands to find messenger RNAs with the same sequence, and this leads to RNA cleavage. Now, what was completely unexpected at this point was the discovery that this is actually a mechanism that cells use to regulate their own genes. So very soon it was discovered that there are actually genes that are encoded in the genome that are functional RNAs. So these are not protein coding genes. These are genes that simply code for a RNA. Once this RNA is transcribed, it actually base pairs with itself and undergoes a couple of conversions and is exported into the cytoplasm. This is called a pre-microRNA. Then once these pre-microRNAs are bound in the cytoplasm by dicer, these are just called simply microRNAs or miRNAs. These are cut into pieces and bound by the RISC complex, and then a search is carried out for any messenger RNA that has a similar sequence. Now, two different things have been found to happen. So these endogenously encoded genes, microRNA genes, actually don't always perfectly base pair with their target. And so if they do not perfectly base pair, it's been found that that just inhibits the translation of those messenger RNAs. If they do perfectly base pair, they actually cause the cleavage of the messenger RNAs. Now, it turns out that these microRNA genes usually affect quite a number of different messenger RNAs. Usually the binding site for a particular microRNA is actually found in the 3' untranslated region of messenger RNAs, and these are generally not a perfect match. So one microRNA might actually inhibit the translation of a whole group of messenger RNAs that have that particular target sequence in their 3' UTR. I just want to clarify the terminology. So microRNAs are the ones that are encoded in the genome of the cell, whereas siRNAs are these exogenous RNAs that are produced due to experimental conditions. siRNAs are also sometimes produced from viruses. Now, it has also been discovered that siRNAs and microRNAs are not only a mechanism to regulate messenger RNAs post-transcriptionally. This is certainly an important component of what they do, but it's also been found that if you have a microRNA or siRNA that is targeted against a particular sequence, that in fact the gene itself in the genome can also become transcriptionally silenced. So this involves a whole separate complex called RITS. RITS stands for RNA-induced transcriptional silencer. And RITS binds to these siRNAs or microRNAs and actually goes to the nucleus and finds a similar sequence in the genome. Once it finds that similar sequence, this RITS complex actually will methylate that region of the DNA and thus inhibit its transcription epigenetically. So I'm going to finish up by just pointing out a few of the important things about RNA interference. So it's really thought to be this ancient mechanism of protection for cells to protect against viruses and also to keep transposons silent in the genome. However, this system has been co-opted by the cell itself to regulate its own genes. And so microRNA genes regulate a lot of different messenger RNAs that are normally produced in cells. This mechanism has also been really important in experimental genetics, and it's relatively easy to actually inhibit messenger RNAs by RNA interference. This is often called knocking down a gene. And then finally, there has been some exploration of using RNA interference to actually treat human diseases.